So good day, ma'am. And we are group number 5, and we will be discussing about animal development. So, so what must we know with this discussion? So we will learn about what is the specific events that is occurring during when a sperm makes contact with an egg cell and fertilizes it. And we will be discussing about the concept of the cleavage, the gastrulation, and the organogenesis. Also, the structures derived from each germ layer. So, going to slide number 3, we can see here a a series of steps which I will be discussing. But first, let me have a brief explanation of what happens after before these events. So we all know after an intercourse, millions of sperm cells are released from the male and travels its way into the female's vagina. And as it makes way to the uterus, which is open during the ovulation stage of the woman, which was also discussed by the previous group, the group 4, is where the sperm cell finally makes its way to the egg cell. But before that, some of the egg cells, the sperm cells, are actually killed because of its acidic environment. However, some of the sperm cells, here as you can see here in my drawing, it's not a very good one, all of the sperm cells are protected by a specific kind of fluid of which enables it to swim safely until it reaches the egg cell. Now, during the ovulation stage, like I stated before, the uterus is open for them to go to the fallopian tube. Now, here, as you can see here, some of the sperm cell may travel to the fallopian tube without the egg cell. And those who travel to the fallopian tube with the egg cell are the ones obviously are going to fertilize the egg. So, where's the egg at the moment? What's happening to the egg cell? So, in actuality, it says that the egg cell is actually being pushed by the cilia found in the fallopian tube, which enables it to move towards the uterus in order for the sperm cell to locate it easily. So as you can see here, it's pushing the egg cell towards the uterus. So here we come to slide number 3. As you can see here, the series of steps are explained. Number 1, the sperm binds the receptors in the xenopellucida, which is the extracellular matrix of the egg. Also, refer to slide number 4. We can see here the egg-shaped kind of cells, referred to as the follicle cells, and also it is referred to as the corona radiata. Okay, the corona radiata. And these are the outermost matrix of the egg. It is the first thing that we are able to see of the egg cell. And inside it, as you can see here in slide number 4, there is a small region of it's the outer membrane of the cell. So before the sperm cell actually reaches and fertilizes the egg, it must first penetrate the zona pellucida, the outer membrane. So how would the sperm cell be able to penetrate and fertilize the egg cell through the zona pellucida? Well, there's a acrosomal reaction, as we say it, as you can see in number 2, the second step. It says that the acrosomal reaction is where the sperm cell releases digestive enzymes, of which enables it to digest to penetrate through the zona pellucida, the outer membrane, and finally reach the surface of the inner membrane. So I will be discussing about the polyspermy after the fertilization of the egg. So finally, the sperm cell finally reaches its way towards the egg cell and fertilizes the inner membrane. When it fertilizes the inner membrane pushes the sperm cell inside and fuses itself, and then the egg cell is finally considered as going to be fertilized. Okay, the first sperm cell finally got through the journey. So what happens now? This is where we call a cortical reaction is about to occur. So, in a gist, the general purpose of the cortical reaction is where a fertilization envelope, as we call it, a fertilization envelope covers the whole egg cell, like here. It hardens, the surface of the egg cell hardens, of which prevents the other sperm cells from fertilizing the egg cell. Now, as I've read in an article in www.bio.edu, it states that this happens because when two sperm cells tries to fertilize the egg cell, it is considered as something lethal and could ultimately kill the zygote, which is why it is important for polyspermy to happen, which is, in a concept wise, like I said before, the prevention of two sperm cell fertilizing a single egg cell. So here we can see a fast polyspermy and a slow polyspermy. A fast polyspermy lasts for almost like a tenth of a second, again as stated by the bio.edu, and a slow polyspermy lasts for about 10 seconds or more. So, as a summary for this overall slide number 3, the first sperm cell, have to penetrate the zona pellucida, the outer membrane of the sperm cell, the outer membrane. It must first penetrate the zona pellucida in order to get to the inner membrane and fuse with the egg cell. So, acrosomal reaction, the head of the sperm cell develops a digestive enzyme and penetrates the zona pellucida and finally fuses with the egg cell. And what happens there is it releases calcium ions, pushes sperm cells out of the way in order to prevent it from preventing more than one to fertilize the egg cell. And finally, the surface of the egg cell hardens, preventing the latecomers, the ones who are about to just see the egg cell, to unable to enter the egg cell finally. So that is the overall gist of the fertilization journey of the sperm cell to the egg cell. Now, what happens inside after the fertilization, this is where the genetic codes finally starts to come. Your traits are finally decided, your gender, your traits, and all of the things that we have discussed, the mitosis, meiosis, from the basic biology that we have discussed in our elementary classes. So what happens after this? What happens when the zygote is finally ready to go to the uterus for further development in 9 months and finally be a living being? Well, first, it has to go through a process called the rapid mitotic cell division. This is where the zygote is finally in transition, as we can see here in slide number 5, a transition of the zygote to the embryo stage. So, I also have another diagram here. So what does the cleavage actually means? It is where the zygote has to develop in a rapid time and become a embryo, but it doesn't have any time to grow. As it says here, the zygote cytoplasm partitionate itself into smaller cells. So it doesn't really grow into an embryo. It first divides itself into smaller pieces. And as you can see here, another illustration of mine, it's not that good. So this is the zygote. It first divides itself into 2, to 3, to 4, and so on, until it finally becomes a morula. A morula is a translation for a mulberry, where it's actually true. You can Google the fruit mulberry, and it really resembles the state of which we are in this part of life. And the parts here goes through more process and divides itself more. So there will be a time where it will compact itself, so compact that there will be more spaces here. And that space is called the fluid that is called the blastula. You can refer to the slide number 5. There are pictures shown below, right here. That's slide number 5. And the overall process of this will be further discussed by my next group mate. So, that is my report. Thank you for your time, ma'am.
Gastrulation is the process by which a gastrula forms from a blastula. A gastrula has 3 embryonic germ layers. Ectoderm as the outer layer, mesoderm as the middle layer, and endoderm as the innermost layer. In this table, you can see the parts of the following parts in each of every layer. This is the process of gastrulation. In the first picture, you can see that a blastocyte reaches the uterus, and after that, it will fertilize for 7 days. And after fertilization, there will be an appearance of maternal blood vessels around the blastocyte. On the third part, you can see that there are extra embryonic mesoderm cells that are appearing for it is on its 13th day of after fertilization, and it starts to develop as and will turn slowly into a gastrula. And the last part will be a perfect formed gastrula, wherein it has the 3-layered embryonic germ cells that are completely and has 4 extra embryonic mesoderms extra embryonic membranes, which are amnion, chorion, yolk sac, and alontis.
There are 3 germ layers in the early development of the animal embryo that is formed during the gastrulation: the ectoderm, mesoderm, and the endoderm. The ectoderm layers is the outermost layer of the embryo. It forms the skin, sweat glands, hairs, nails or hooves, eyes, mouth, and teeth. It also forms the nervous system, that is the brain and the spinal cord. Mesoderm, the middle layer between the ectoderm layer and the endoderm layer, plays an important role as it forms the skeletal system, muscular system, circulatory system, and the excretory system, lymphatic system, and the reproductive system. The endoderm is the innermost layer of the embryo. It forms the lungs, livers, pancreas, as well as the thymus, thyroid, and parathyroid glands. It also forms the lining of many organ systems, such as the respiratory system, digestive system, urinary system, and the reproductive system. The frog embryo is a ball of cells with a fluid-filled cavity. The cavity is the blastocele, and the embryo is in the blastula stage development. The blastula contains a large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The 3 colors represent the 3 tissue layers. Yellow indicates the endoderm, red indicates the mesoderm, and blue indicates the ectoderm. At the beginning of gastrulation, a few surface cells called bottle cells move into the interior embryo, followed by other surface cells. The movement of cells into the embryo create a dorsal tip, which sheets of cells continue to move inside at the same time. The ectoderm extends around the embryo surface. A cavity called arcentrion forms while the blastocele shrinks. The arcentrion is surrounded by endoderm tissue. As ectoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surfaces follow them, creating the ventral lip. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm lines the inside, and endoderm lies between the two.
Gastrulation in a chick embryo. By the end of the 2nd week, yung epiblast and hypoblast ay makuform. Ini-establish nito yung 3 embryonic germ layers, which are the ectoderm, mesoderm, and endoderm. Sa process ng gastrulation. 15 days after ng fertilization, may mapoform na makapal na structure sa gitna ng epiblast, which is the primitive streak. Dumadaan dito yung primitive groove. Pag na-form na yung primitive groove, yung epiblast cells ay magmumove papunta dun sa streak, at magsislip ito sa interior ng embryonic disc in a process of invagination. Yung unang cells na i-invaginate is yung primitive group na mag-i-invade sa hypoblast, wherein mapapalitan ito ng bagong cell layer which is yung endoderm. Yung ibang epiblast cells at yung mga bagong na-form na endoderm na matitira dito ay magpo-form sila ng germ layer which is yung mesoderm. Yun yung mga nasa gitna doon. Pagtapos na yung formation ng endoderm and mesoderm, yung epiblast cells, hindi na siya pupunta dun sa primitive streak. Yung mga natitirang epiblast cells, which is the ectoderm, nagpo-form naman sila nung third layer sa ilalim na yun. Tapos, yung process ng gastrulation is complete na pag yung tatlong germ layers ay nakompleto na sa embryonic disc. Organogenesis. Ito yung pagdevelop ng organs ng animals or ng plants. Notochord naman, ito yung major skeletal element sa pagdevelop ng embryo. Neural plate is the developmental structure na nagsuserve as the basis for the nervous system. Neurulation, which includes the transformation of the neural plate to the neural tube. And lastly, yung somites, ito yung division ng body ng animal or ng human.
Amniotic embryo is developed in a fluid-filled sac within a shell or uterus. So that sac is commonly called as the bag of water or amniotic sac. So it is full of water because that fluid, it is also called as amnion or amniotic fluid. It has the responsibility to protect the embryo from its environment. It also prevents dehydration, cushion, and mechanical shock. So, meron din tayong part na tinatawag na yolk. Nandun lahat ng nutrients. So, the nutrients of the egg. So, ayun yung tawag sa kanya. Next is we're going to discuss about patterns of development. So, sa patterns of development, may 3 words tayo na kailangan tandaan. Ito yung cytoplasmic determinants, induction, and totipotent cells. So, what is cytoplasmic determinants? So, cytoplasmic determinants is a chemical signal such as mRNAs and transcription factors influence patterns of cleavage. So, sa cytoplasmic determinants, tawag sa kanya ay chemical signals. Tapos, ini-influence niya yung patterns ng cleavage. So, kadalasan siguro, nagkakaroon ng changes dun sa patterns ng cleavage ay dahil yon sa cytoplasmic determinants. Ang next natin is induction. So, what is the difference between cytoplasmic determinants and induction? So, induction is the interaction among cells that influences their fate, causes changes in gene expression. So, diba? Induction, it also causes changes sa gene expression naman to. So, it is happening within the cell. So, it has happening in the cell. So, kung ano yung relationship ng each cell, pag nagkaroon ng changes doon, pag nagkaroon ng changes sa gene expression, dahil yon sa induction. So, the next is totipotent cells. So, what is totipotent cells? It is capable of developing into all the different cell types. So, yun yung totipotent cells. So, all cells of mammalian embryos are totipotent cells until the 16th cell stage. So, yung lahat ng mammalian embryo ay kasama pa sa totipotent cell hanggang 16 cell stage.